Welcome to the video on dissolution limits and the safe space approach. Many of the viewers may be finding this topic as a very new topic to them, and these terminologies may be new for many of the viewers. This video will be very informative video and very interesting video for those who wants to learn about the dissolution limits and who wants to know what is the concept of safe space approach for dissolution limits. As we know, dissolution is the performance test for pharmaceutical formulations, mainly for the oral formulations and other formulations for which the dissolution testing is done. Dissolution testing is a in vitro testing which provides information regarding the behavior of product in vivo. Dissolution testing gives information about bioavailability and also about the bioequivalence for the formulations for which dissolution testing is required. So let's start with the video and let's understand these concepts. So safe space approach. We know that dissolution limits are required to be given in the drug product development, in the commercial phase as well, and dissolution testing is done in all phases of the product development from research phase till the life cycle. It also to be done in the scale up and post approval changes. Dissolution testing is also required when your stability testings are performed. Dissolution testing is the test which ensures the bioequivalence, bioavailability and patient safety with flexibility. So we know that dissolution specifications are set or acceptance criteria is set based on the clinical trial batches and the bioequivalence batches. So is it too strict to base dissolution limits on just one batch? The answer will be no. But in practice, for generic formulations and formulations which are innovative, the clinical trial batches may not be more, and we set the acceptance criteria based on the clinical trial batches, clinical study batches and the bioequivalence batches. Small variations in the manufacturing or testing may lead to unnecessary batch rejections, resulting in delays and cost escalations. This is the main problem when we are setting the dissolution limits based on just one clinical batch or bioequivalence batch. For example, I will give you, there are many products which are already available in the market. These are the formulations which have different release rates and may have different specifications, but still these formulations are bioequivalent to the reference formulation if you consider the generics, or if there are some differences in the dissolution profile of different batches, still they are bioequivalent, or still they may prove the bioequivalence. So for example, for your easy understanding, I am giving you that one batch is having 100% dissolution in 10 minutes. Another batch or another product, you can say, for the same formulation is having 100% dissolution in 15 minutes. And one another you can consider with 100% dissolution in 30 minutes. So we cannot say that these formulations are bioequivalent or bioinequivalent. This depends on the product, the molecule, and the in vivo behavior of the formulation. So if you have performed biobatch with the release rate of, let's say, 100% release in 15 minutes, and you set the dissolution criteria based on that, then if your batch is showing 100% resolution in 30 minutes, so you will be going to reject that batch because you have set the acceptance criteria considering the bioequivalence batch, though that batch may be bioequivalent. So this is the main area of focus for this video, and that's why the small variations in the manufacturing or testing may lead to unnecessary batch rejections, resulting in the delays and cost escalation. So as we know, in the quality by design approach, we have the knowledge space, design space, and the control space. So this is also applicable for the dissolution limits. If the dissolution of the batch goes beyond certain boundaries, then only it may prove that the batch is bioinequivalent or it will fail in the bioequivalence. But if the dissolution specifications are varying in that specific range, then these batches are the bioequivalent batches. Coming to the introduction for safe space approach. So the safe space approach offers a range of dissolution limits like upper and lower boundaries instead of rigid single value limit. For example, if you are given a Q point for IR formulation like 80% in 15 minutes or 30 minutes, so you can give some ranges and you can have the understanding about the ranges for dissolution specifications. If the batch fall within that specification, it will be bioequivalent. So this approach ensures bioequivalence and safety while accommodating the minor variation in the manufacturing so that your batch rejections will be less. Coming to the problem with rigid dissolution limits. So the practice is that dissolution limits are often based on the single bio batch. As I have discussed earlier, single bio batch is there and based on which we set the dissolution limits. The impact is that minor variation in the manufacturing or testing can result in the batch rejections. And consequences of this problem is delays instead of cost and potential risk to patient to access the medications. Some medications are very limited in the market. Some medications or formulations are manufactured by only some of the companies. So if the batches are rejected, then market shortage for that formulation or product may happen. So what is the safe space? So it is a range of in vitro specifications for key quality attributes like dissolution. And its purpose is this range allows for small differences across batches while still ensuring they remain bioequivalent and safe for the patients. Boundaries are defined by upper and lower limits for key attributes like dissolution. And all batches within this range should be bioequivalent despite of the minor differences. So this is the safe space. So how this approach works? Instead of identical dissolution results, establish a range where small variations are acceptable. And the outcome of this will be the product remains bioequivalent and safe for patients even with minor differences in the production. See, dissolution variation may happen because of many of the reasons, which may be the raw material differences, minor variations, manufacturing variations, testing variations, storage variations, or maybe because of the variations in some of the parameters. But all these variations may or may not lead to variation in the dissolution, and still if variations are there in the dissolutions which are minor, may not lead to bioequivalent product. Dissolution testing evaluation. So the role of dissolution testing is the quality control. And nowadays, dissolution testing is becoming a surrogate for certain oral formulations, bioequivalence testing. So this is the dissolution evolution. Ensuring the bioequivalence. So current method is that all commercial batches must exhibit similar dissolution profile to the bio batch. And the main challenge of this approach is that strict limits can lead to batch rejection due to minor variations, delaying the productions. So now come to the safe space versus strict limits. Strict limits based on the single bio batch, often leading to rejections due to minor manufacturing or testing variances or variations. And safe space establishes an acceptable range accommodating real-world variations while maintaining the bioequivalence. So here I want to mention one thing, that when you are working onto the formulations, and whenever we prepare the batches for pivotal bio studies, we have generally the strict limits for API, strict limits for the processes, and the API CMAs like particle size distribution. But when this product goes from design stage or exhibit stage to the commercial stage or commercial phase, the variations or differences may come into picture regarding the API particle size, regarding the scale of manufacturing, regarding the site. So all these variations will lead to minor variations in the dissolution, and that's why safe space establishes an acceptable range accommodating the real world variations while maintaining the bioequivalence. What is the importance of clinical relevant specifications? So these clinically relevant drug product specifications are based on the in vitro and in vivo links. Resolution profiles within the safe space are considered bioequivalent, ensuring better patient outcomes. The reason why the safe space approach matters, because it is patient centric, it ensures minor differences in the resolution which are acceptable, focusing on patient safety and access to affordable medications. And its long-term benefits are assures quality, supports regulatory flexibility, and reduces unnecessary clinical studies. These safe space resolution specifications are also related to the clinical outcome of the batch. The safe space approach offers more flexible, realistic solution for setting dissolution limits while ensuring bioequivalence. This approach provides patients with safe, effective, and affordable medications without unnecessary delays or increased costs. So now uh, you should try to relate these things for dissolution testing in QC. We should be discriminatory, we should be biorelevant, and then it should be clinically relevant. And from starting of the product from research phase till the commercial, the dissolution limits plays great role. Also, there may be minor variations within the batches or within the different packs of the same batch, also within the different storage conditions. So always have an idea about this safe space approach, considering the clinical relevance and the bioequivalence. So this is the information regarding dissolution limits or specifications and the safe space approach. And I hope this video will help you to understand this new or less known information about dissolution specification based on the safe space approach. So the example may be like, if the batch has the dissolution of, let's say, 80% within 15 minutes, or 90% within 15 minutes, or 100% within 15 minutes, or at 15 minutes, all the batches or the batches with all these release or percent dissolution will be bioequivalent. Or let's say the dissolution specification of 70 to 90% will give rise to bioequivalent batches. So this is the main approach for setting the dissolution limits. Thank you for watching the video, and if you have any comments or questions, please feel free to ask and I will be happy to answer your queries. Thank you.